Welcome to this episode of Mentors at Your Benchside, the podcast giving you advice, tips, and tools for getting the most out of your research. I'm Jason Rogers, and today I'll be talking to you about the top uses of microbes in biotechnology. Biotechnology is the use of biological organisms in technological processes. It is almost as old as civilization itself, although it wasn't called biotechnology until the 20th century. Far from abandoning it in the 21st century, we are developing new uses for biological organisms. Number 1. Beer, bread, and wine. Biotechnology used for food and drink production is called yellow biotechnology. Simple fungi in yeast forms were likely the first domesticated organisms used to make edible products through the process of fermentation. The consumption of glucose by yeast yields a byproduct of ethanol and carbon dioxide, which can be exploited for generating bread, beer, and wine. Likewise, the popular hipster drink kombucha and fermented cabbage kimchi are made through the fermentation of yeast with lactic acid bacteria and acetic acid bacteria. The most famous yeast is brewers' and bakers' yeast, Saccharomyces cerevisiae. It was first used by ancient Egyptians to make beer. Even the pyramid builders had beer rations. S. cerevisiae are harmless and easy to culture. Its culturing method was eventually refined by Louis Pasteur. In 1996, the genome of baker's yeast became the first sequenced eukaryotic genome. It's no wonder that it was the first eukaryotic organism used as a model for the production of various metabolites, from isopropanol to methanol. See our articles about working with yeasts. Beyond yeast genetics, yeasts are used as a model to study protein-protein and protein-nucleic acid interactions, both of which are handy in drug discovery, with the help of various hybrid assays such as 1 hybrid, 2 hybrid, and 3 hybrid. Number 2. Milk products. Fermenting milk using microorganisms is as old as domesticating herd animals, which began around 14,000 years ago in the Fertile Crescent. Milk is fermented by lactobacillus. However, some cheeses, like blue cheese, are made with a fungus, Penicillium. Similarly, certain dessert wines are made from the noble rot of the fungus Botrytis cinerea. Number 3. Antibiotic Production. Red biotechnology is the use of biotechnology in the medical and pharmaceutical industries. It's called red because of the association with blood and symbols of medicine, the Red Cross and Crescent. Fungi are most commonly associated with cheese and wine production. However, they have a long history in traditional medicine, as mouldy bread, overgrown with the fungi Penicillium and Aspergillum, would regularly be applied to wounds for their antimicrobial effect. The jump from antimicrobial compounds to the antibiotics we know now was ushered in by Scottish scientist Alexander Fleming, who accidentally discovered Staphylococcus growth suppression by penicillin mold in 1928. However, it took another 12 years and a world war to start mass-producing penicillin using deep fermentation techniques. Number 4. Restriction enzymes. It had been known since the 1950s that certain bacteriophages demonstrated poor growth on different bacterial strains, but the reason was unknown. In 1970, US researchers characterized an enzyme from the bacteria Haemophilus influenzae that can cut DNA in specific places with the purpose of protecting the bacteria from foreign DNA. The enzyme was HIND3, the first type 2 restriction endonucleases, or restriction enzymes that scientists adore. Today we know more than 3,000 restriction enzymes recognizing 230 DNA sequences. Number 5. Protein production. Protein production in E. coli was the new stage in biotechnology because it used recombinant DNA technology instead of traditional selection techniques. The first human protein produced in E. coli in 1978 was insulin, followed by human growth hormones. Number 6. Eukaryotic viruses. Using E. coli to express eukaryotic proteins has challenges. Firstly, the proteins expressed in bacteria lack post-translational modifications that decorate many of the eukaryotic proteins, from glycosylation to assembly of protein complexes. Secondly, E. coli reluctantly produces large proteins common in eukaryotic cells. No wonder the first therapeutic proteins expressed in E. coli were relatively short and simple. For more complex proteins, such as antibodies, the production shifted to eukaryotic cells, Chinese hamster ovary and human cell cultures. But even in eukaryotic cells, biotechnology cannot work without the help of microbes, example viruses. While the expression vectors in E. coli are based on bacterial plasmids, the vectors for eukaryotic cells are viral vectors. The first proof of principle was a vector based on SV40, simian vacuolating virus 40, that infected a cell culture of monkey kidney cells. Number 7. Phage display libraries. It would be fair to say that molecular biology started with bacteriophages. Aside from the discovery of restriction enzymes, the enzyme that joins the ends of the recombinant DNA molecules, T4 ligase also comes from a bacteriophage. But maybe the biggest phage contribution to biotechnology is phage display libraries, PDLs, developed in 1985. In PDL, a gene encoding a protein of interest is inserted into a phage coat protein gene, causing the phage to display the protein on its outside. PDLs are used in the studies of protein-protein, protein-peptide and protein-DNA interactions. PDLs are useful in vaccine and drug discovery. Number 8. Microalgae. Blue biotechnology uses sea resources to create products and industrial applications. The leading application of blue biotechnology is producing renewable bio-oils with photosynthetic microalgae to replace oil extraction. Working with algae is not much different from working with other microorganisms. To genetically modify your microalgae, use the familiar method of DNA introduction into a cell transformation. Number 9. Agrobacterium to Mafan. Green biotechnology is biotechnology involving the genetic engineering of plants. It's based on a neat trick by the microbe Agrobacterium to Mafan. It has a plasmid, Ti, that transfers some of its genes into the plant genome. The transfer requires only tDNA border sequences, and you can insert foreign DNA between them that will be integrated and expressed in a plant genome. Several cereal crop plants were modified using this method, as well as HeLa cells. Number 10. CRISPR. Last but not least in the history of biotechnology is the use of CRISPR. Of course, it's not a microbe, but rather a bacterial mechanism that's used to engineer organisms. CRISPR was discovered in 1987 but has gained considerable traction in the last decade. In short, bacteria have no immune system, so they evolved a mechanism to recognise and fight off bacteriophages should the bacteria encounter the bacteriophage a second time. Each time a bacteria is infected with a bacteriophage, the bacteria stores a bit of DNA from the bacteriophage genome. Using the bacteriophage DNA sequences as a guide, the Cas9 enzyme, encoded by the bacteria, recognises similar DNA sequences, i.e. a second infection by the same type of bacteriophage, and destroys them. Scientists recognise that this same mechanism could be used to introduce stable changes into the genetic material of eukaryotic cells as well. So that's it for all the amazing uses of microbes in biotechnology. Check out the episode description for links to related articles and resources, and don't forget to subscribe to the podcast to get more help and advice from mentors at your Benchside.